Today's assembly is the second lecture of the 5th Yen Kwo-Yung Lecture in Life Sciences, jointly organized by New Asia College and School of Life Sciences. We are pleased to have Sir Richard J. Roberts, 1993 Nobel Laureate in Physiology or Medicine, sharing with us on why I love bacteria, 為何我鍾愛細菌. We are also pleased to have Professor Henry Wong, our college head, to introduce the speaker and to be our moderator. Now may we invite Sir Richard and Professor Wong to come up to the stage. Sir Richard, please. Professor Wong, please.
Sir Richard, honorable guests, fellow alumni, colleagues, and students, good morning. It is always a pleasure to introduce a distinguished scholar at the College Assembly. The year 2019 is an extremely important year of New Asia College because it marks the 70th anniversary of the college, as well as the 5th year of Yen Kwo-Yung Lecture in Life Sciences. This year, we are honored to have Sir Richard J. Roberts, the 1993 Nobel Laureate in Physiology or Medicine and Chief Scientific Officer of New England Biolab, to share with us his insightful ideas and thoughts. New Asia College and the School of Life Sciences have co-organized the Yen Kwo-Yung Lecture in Life Sciences since 2015 as a result of a generous donation to New Asia College from the family of Dr. Yen Kwo-Yung, founding chairman of the Department of Biology of the college. The lecture series aims to broaden the knowledge of life sciences among staff, students, and the general public, particularly in its value and applications in modern society. Today, we will have the second talk here in this auditorium, and Sir Richard will speak on the topic, Why I Love Bacteria. Sir Richard studied chemistry, physics, and mathematics in the University of Sheffield. After obtaining his doctorate degree, he became a postdoctoral fellow at Harvard University. In 1993, Sir Richard and Philip A. Sharp were jointly awarded the Nobel Prize in Physiology or Medicine for their discoveries of split genes. Since the early 1990s, Sir Richard has been working as research director at New England Biolab, a molecular biology reagent company located in Beverly, Massachusetts. His current research focuses on DNA methylation in bacteria and the use of bioinformatics to discover new bacterial gene functions. Last but not the least, may I also take the opportunity to pay tribute to the donors, Dr. Yen Kwo-Yung and his family, for the endowment, which is most vital to make these meaningful academic lectures possible. Thank you. Now may I invite Sir Richard to deliver the lecture, Why I Love Bacteria. Sir Richard, please.
Thank you. Well, thank you. I'm going to talk to you today about bacteria. I love them. I absolutely adore them, and I hope that by the end of this talk you will too. I think bacteria have had rather a bad name over the years, and I'll explain why. But I want to start off by talking a little bit about the variety of bacteria that are out there. You find them in every possible part of the earth, and some of them are extremely interesting from many points of view, and I will end up talking about bacteria and humans. This is an abbreviated tree of life. If you see up here, these are the bacteria, all coming up around here. Down here, this is humans. If you want to see how far away we are from bacteria on the tree of life, you have to come all of the way down. This is more or less accurate as to how far away we are. This is maize, this is a plant, and this is man. They're pretty closely related by comparison with the bacteria. And bacteria, of course, were here long before we were. In fact, almost all of the genes that we have in our bodies originated from genes that were in bacteria or in archaea, which is the other major branch of, the other major branch of life shown over here. The archaea, I will show you some examples of. These are not bacteria. These have some resemblance to bacteria, but they're also rather close to eukaryotes. This is a very interesting bacterium, Anabaena St. John's. It's typical of the bacteria that live in the oceans. And one of the reasons that we have oxygen in this planet that we can breathe is because the original bacteria, like the cyanobacteria, of which Anabaena is a representative, were anaerobes. So they only wanted an environment in which there was no oxygen. And then they made the mistake of metabolizing whatever was hanging around, and they started to produce oxygen. And in the long run, they actually almost polluted themselves out of existence, something that we're in danger of doing at the moment. They didn't know better. I think we do, and we have to make sure that our political leaders make sure also that we do not suffer the same fate as many of these bacteria. The Anabaena were smart in one sense. They have little cysts that they form in which these are anaerobic chambers in which they can continue to grow. Here's another very interesting bacteria, rather beautiful, look like little mushrooms. These you find in the forests, and these are bacteria that typically are responsible for rotting wood, for getting rid of trees, getting rid of all the debris that comes from the foliage of trees. We find these all over the place. They're tiny, of course, you don't see them, but you see their effects. Every time you see a tree rotting, they're usually full of myxobacteria. Gosh, this doesn't change the slide so easily. Okay, this is a close-up of one of them. They're rather nice. They have quite big genomes, and they do a bunch of interesting things, but I'm trying to avoid telling you about too much science in this talk. I want you to rather come away with the idea that there are a lot of extremely interesting bacteria out there, and they're absolutely vital for life. Here's another interesting bacterium. It's a spiral bacteria. It has flagella, these little appendages here, but it's got something inside it. You can see right here, there is an iron core, the element iron, and that is a little magnet. And it turns out that this bacterium orients itself according to the Earth's magnetic field, and in the northern hemisphere will swim north looking for food, and in the southern hemisphere they swim south looking for food. They're really quite sophisticated. Who would have thought that a simple bacterium would have somehow built its own magnet to help it guide its way around? I see this, okay. Now I want to talk now about another extreme environment that is out there. You probably know that there are many underwater volcanoes on the planet. If you look down in the Atlantic Ocean, there's a ridge that runs down there, and these are littered with small volcanics. We call them black smokers. And these are pouring magma out of the Earth's core, and of course, as it comes out, it's extremely hot. But it soon cools off because the seawater around it is rather cold. And so you get this big thermocline, a big thermal gradient in which you find organisms living. Now, these black smokers don't last forever. They're not continuous. They tend to erupt, just like a volcano does, and then they die down. But the life forms get to these volcanoes very quickly. We don't exactly know how they do it most of the time, but there's quite a variety of life forms there. Over here on the right are some things that we call tube worms. These organisms are really just a big shell. They have a eukaryotic worm-like structure inside them, but what they live on is actually hydrogen sulfide. And they have inside them colonies of bacteria that can live on hydrogen sulfide. I see the slides like to advance 2 at a time. I'm sorry about that. So this is a close-up of the tube worm. And you can see they have this casing, the white casing, and then the red is the actual tube worm. This is not working as well as one might like. Let's try it this way. Another interesting environment are the thermal hot springs that you find. You probably know about the geysers that were originally found in Iceland, in which, again, you have not exactly volcanic eruption, but something rather similar to this. One of the great sources of these particular kinds of geysers is Yellowstone National Park in the US. I would advise everybody, if they go to the US, try and get to Yellowstone. It's an amazing place. It has a big lake that's at normal temperature, but all around it, there are hot springs coming up. There are hot springs everywhere. And these hot springs are absolutely full of life. These green organisms here, a variety of bacteria, some cyanobacteria, some are others, and they live in a very narrow temperature environment, which they love, and they hang around. Over here, there are a bunch of organisms that produce and take iron out of the rock. And so you end up with these beautifully colored arrangements around the geysers and around the hot springs, and these are all bacteria that are growing there. Now, I want to switch, and I want to talk about humans and bacteria. And just in case you're not aware of it, if I were able to kill all of the bacteria that are living with you, which you might think was a good thing because, of course, they cause disease of one sort and another, but in fact it's not a good thing because you would die. Your bacteria, the ones that live with you, that crawl all over your skin and are in your gut and in your hair, in every body part, these are absolutely essential for your life. Now, you might not think that, because what happened was that the microbiologists, when they first discovered bacteria living with humans, they were interested in the bacteria that cause disease. And I'll show you a little about this. And what happened was that there was just this massive amount of scientific work done looking at pathogenic bacteria, looking at all the bad ones. And so most people came away after about 100 years of research thinking that all bacteria were bad. In fact, it's exactly the opposite, as I will show you in a moment. Now, when we look at the number of cells that are present, the number of human cells that are present, They're of the order of 10 to the 13. That's a pretty large number. You've got a lot of human cells. There's at least as many, maybe 10 times as many bacterial cells that live with you. So there's actually more bacteria living with you than there are human cells. If you look at the number of strains, typically a human will have in excess of 20,000 strains, maybe as many as 50,000 different strains of bacteria living with you. And they will vary in where they like to live and what strains are present. In terms of the amount of DNA, the human genome is about 3 billion base pairs long. The total length of all the DNA in the bacteria with you is about the same. But the human DNA is full of introns, these things that I discovered that I won the Nobel Prize for. So the actual number of genes is pretty small. The number of regions in the human genome that code for proteins is pretty small, maybe 23, 24,000, something like this, not that many. But when we look at the bacterial genes, tremendous variety of genes, and more than 1 million bacterial genes that are helping keeping you alive. This just gives you some idea of the microorganisms that are present in humans. So when we look on the skin, you get a lot of staphylococci. You often think of Staphylococcus in terms of Staphylococcus aureus, which is rather nasty. In fact, most of the staphylococci that live with you are Staphylococcus epidermidis, and they live in little gullies, little cells, little pores that are present in your skin. And these are very important. They're not pathogenic. They help keep your skin looking good. Also, many others. In the mouth, you have a lot of organisms that live in your mouth that very often are responsible for starting the digestion of the food that you eat. You know, we eat food to keep us alive, but it is the bacteria that actually digest the food and convert it down to the metabolites that ultimately provide our source of energy. So without the bacteria, you would have a hard time dealing with all the food that you eat. In the respiratory tract, there are also large numbers of organisms living all down through the lungs. Of course, occasionally tuberculosis will live there, but there are other bacteria that live there too. And so on, coming on down. There are many, many different genera of bacteria, and almost all of them serve some useful purpose for our lives. Now I want to tell you why bacteria got a bad name. And that started off with a man called Robert Koch. And he was one of the foremost bacteriologists who was really looking to see why people got sick. If you go back into the 19th century, bacterially acquired infections were really a big deal. Even today, they're still a big deal. Despite the fact that we have antibiotics and we have vaccination, there are many diseases that essentially should be eliminated, even though the anti-vaxxers do their best to stop them from being eliminated. But the early microbiologists were really focused on these bacterial infections. And so you see anthrax was a big problem caused by a bacillus, suppuration by staphylococci, gonorrhea, of course, this disease you don't want to have to go home and tell your family about. This was found to be caused by a Neisseria gonorrhea, which is still fairly widespread. And coming on down, tuberculosis, and you'll see many of these early ones were discovered by Koch. He understood, figured out what these were doing. Coming all the way down here to 1906, we're down to whooping cough, bordetella, syphilis, caused by a treponema. While it is true that these are dangerous bacteria, if I were to look in your gut, or look on your skin, or look within you, I would find that many of these organisms were present. But you would find that they're present in exceedingly small amounts. And they're present in small amounts because the normal gut bacteria, the normal skin bacteria, have found a way to stop them from growing. And this is something that's really worth remembering, that most of the bacteria that live with you are digesting your food, or they're trying to keep you healthy. In fact, they don't like it when you get sick, because we are the home of these bacteria. They've made us their home. And what do when you get a new home? You put an alarm system in. You put a fence around it, and you stop the burglars from getting in. That's what these bacteria, most of the bacteria within us do. These are the rarities, the ones that get away from these control systems. Now, I want to tell you about some of these nasty, so-called nasty bacteria, and I'd like to start by talking about Helicobacter pylori. Helicobacter is a very interesting organism. It causes ulcers. And until, probably 20, 30 years ago, everybody thought that ulcers were caused by too much acid in your gut. And so the pharmaceutical companies starting selling alkali, the idea being that you would solve the ulcer problem. But a man called Robin Warren, in conjunction with Barry Marshall, looked in the gut and discovered that people with ulcers had this organism, Helicobacter pylori, growing there. And Barry Marshall did an experiment. He said, I think it is these bacteria that cause ulcers, not acid. And so he actually swallowed some Helicobacter pylori that he'd grown in the lab. And sure enough, in a relatively short period of time, he developed an ulcer. Now, fortunately, the Helicobacter that he swallowed was sensitive to antibiotics, so he could take antibiotics. Lo and behold, the ulcers went away. Robin Warren and Barry Marshall got the Nobel Prize for this discovery. But there's an interesting lesson that goes along with it. When they first made this discovery, nobody wanted to believe them. And in particular, the pharmaceutical companies didn't want to believe them. And they went out of their way to try to show that what they were saying was wrong. And they did everything they could to try to stop this piece of science getting into the public perception. You have to be very careful whenever companies are coming along and telling you stories that might affect their bottom line. Most companies are there to make money, and they don't like anything that stops them, even if the science tells them that it's correct. Just think about global warming and the oil companies. Now, this bacterium, while is rather nasty, you might think this was not good. It causes ulcers. It turns out it can also cause cancer. You can get cancer of the stomach and cancer of the esophagus when these bacteria get out of control. Not usually a big problem, but it can be a problem. And so you might think that it would be really good if we could wipe out all Helicobacter pylori. But as with most stories, there's a silver lining. It turns out that if you have a good Helicobacter infection going, not that it's dangerous, not one that is causing cancer or causing ulcers, but just naturally growing, you do not get asthma. This organism protects you from asthma. If you look in the developing world, that is almost an unknown disease because they're almost all infected. You look in Hong Kong, you look in Singapore, you look in the developed world, we have taken so many antibiotics that typically we have killed all of our Helicobacter pylori. And this is only one of the downsides of taking too many antibiotics. We have gone crazy in the way in which we prescribe antibiotics, in the way in which we make them available over the counter, to the point where now many of the bacteria that we would like to be able to kill, to get rid of when they're pathogenic, they're resistant because we have overused antibiotics so terribly. Lyme disease, I don't know if you have it here in Hong Kong. Maybe you don't. This is a disease that is present in the US. It's present in many Caribbean islands. It's caused by an organism called Borrelia. Borrelia is carried by ticks. When you go out, the ticks that carry Borrelia live on deer. When you go out in the summer, where I live, then you get bitten by a tick. If the tick has a Borrelia infection, transmits this bacterium to you, and you end up with these lesions. You see a big lesion on your arm or your neck or wherever the tick bit you. It takes a few days for this to come up. It's only there for a short time. It's there for about 2, 3, 4 days, and then it disappears. If you catch the circle, this lesion, when it occurs, you can take antibiotics. That will kill the Borrelia, and you won't have a problem. But if you miss this, and you don't get the Borrelia early, it can spread around your body, and it gets into the nervous system, and it gets into the brain, and it can actually cause dementia. It can be a very serious problem, and it is very, very difficult to get rid of it once it's got firmly established. And it's a shame, in a way, because this is Borrelia. Borrelia is this beautiful spirochete. It's a nice spiral organism. Looks rather nice, but can be deadly in the sense that it will cause brain damage, and you will not enjoy the rest of your life if you don't do something about it. I want to tell you another story, an interesting story. This is about Vibrio cholera. You probably have all heard about cholera. Cholera is something that gets into epidemics when you get this organism into the water supply, and it can be very nasty. It's really a serious problem. Turns out that this organism is present in seawater all the time. You and I go out swimming. We swim in the seawater that contains Vibrio. Doesn't usually cause a problem. It mainly causes a problem when it gets into the drinking water supply. So as long as you just get on your skin, it's okay, but you start drinking it, it causes a problem. And this was particularly dangerous in Bangladesh, among other places. They have a lot of problems with it because Bangladesh is a very low-lying country. You get seawater coming into the drinking water rather regularly. A lady called Rita Colwell, who used to be head of the NSF in the US, made a startling discovery. She realized that Vibrio cholera, this bacterium, likes actually to hang out with a larger eukaryotic organism and typically sticks to it. This is a Volvox around here, and the individual Vibrio cells like to hang on to the Volvox. Now, Volvox is quite a big organism, and you can filter it out of water. In fact, the ladies in Bangladesh wear these beautiful saris that are very tightly woven. And if you just filter the water through the sari before you drink it, you get rid of 99.9% of all of the Vibrio. And this is a way of taking water that is contaminated and making it clean. Very, very simple solution, because all of the ladies in Bangladesh wear very nice saris, and they wear these tightly woven saris. Botulism. You've probably heard about botulinus toxin. It's a very dangerous toxin. Its cause comes from this Clostridium botulinum. This is also the toxin that a lot of ladies like to inject in their lips to make the lips larger and apparently more beautiful. It's caused by this very tiny little organism, this little Clostridium here. And I'll tell you something else about Clostridium in a moment, but I will just leave it at this at the moment. It's a dangerous organism. It can cause disease, and yet it has some beneficial qualities, or at least many ladies think it has beneficial qualities. Not the organism, but the toxin that it produces. Typhus is an interesting disease. This is caused by an intracellular bacterium. This is a bacterium that lives inside human cells. And you can see inside the cell here you have these little particles. Causes typhus, which is a very nasty disease. This was something that was a major problem during the First World War, during the Boer War, during early wars. Spreads very easily. Fortunately, resistant to antibiotics. And so this is a disease it's easy to treat with antibiotics. Treponema denticola. This is an organism, a treponeme, that lives inside your mouth. Most of the time, it doesn't cause a problem, but occasionally it can grow out of control and cause a small problem. But it's rather beautiful. Another of these beautiful little spiral bacteria, long, elongated spiral. Okay, I want to talk about Yersinia pestis. This was the organism that caused the plague in the Middle Ages in Europe and decimated the population. Massive killing of people, because the only way you could stop getting it was to go and isolate yourself so that you stayed away from anybody who had the disease. And this has been a constant way over time that people have tried to get away from infectious diseases. You either take the people with the disease and put them in essentially a prison and stop them from infecting everybody, or you go and hide and get away from them yourself. When I was a kid, tuberculosis, TB, was a major problem. And the treatment in Britain, we had sanitaria. Every major city had a sanatorium. And if someone got TB, they had to go and live there for a period of time until either they were clear or they died. It was very difficult to get out of the sanitaria until penicillin came along. Turns out penicillin was the wonder drug for tuberculosis, but is no more, no longer a wonder drug. You can detect it by these buboes. These are little swellings that you can get in your knees, you can get them in the elbows, you get them on your body. Now, you might have thought that this disease would have been wiped out. It's not. There are many cases in the US every year, and usually it's because rats carry the disease, they spread it to humans, and it's quite common in the West, out in Arizona, in New Mexico. This is quite a common disease. And people who get it go and see the doctor. The doctor immediately recognizes these buboes and says, oh, you've got the plague, you've got Yersinia pestis, give you an antibiotic, you're cured. But what happens very often is people have already left Arizona, maybe they're traveling to New York, they're on vacation, and all of a sudden they see this, they go to the doctor. The doctors in New York have never seen this. And so many places where this is common, where you know that it happens and you can cure it easily, and you have to get it early. It's no good waiting, you have to get it early. And so always be careful when you go to doctors. It's better to go to a doctor in your local neighborhood who has probably come across the disease that you have. This is something that happens. This is, you get all this gangrenous tissue on your fingernails, you get it on the toes, and this usually leads to amputation. The only way you can get rid of it is to amputate the limb. This is the bug itself, nice little red bug, nothing special to worry about. But you know, despite all of this, you might think that we're surrounded by organisms that are out to kill us, that bacteria are really there to kill us. And I would say until probably about 15, 20 years ago, almost every microbiologist you spoke to would only tell you about all these bad ones. But despite all of that, I love bacteria. And the reason I love them is because they protect us. They've made us their home. They want us to be well. They want us to be safe. They don't want anything to happen to us. And so they protect us. And we don't know how they protect us. This is one of the great open areas for science at the moment, particularly for biology, for microbiology. It would be very nice to know how the resident bacteria in your gut can stop pathogens from growing. And I'll give you a good example of something in the moment, of how that can happen that we don't understand. But we know that it happens because, as I said earlier, if I look at all of the bacteria that are living with you, you will find that many of these pathogens I've just been talking about are present in your gut. But they don't cause a problem. They're stopped from growing out of control by all the good bacteria. And 99.99% of all the bacteria that live with us are good. They're keeping us safe, they're helping us. And surprisingly, they don't like us getting cancer either. And so we've now found there are many bugs that produce anti-cancer compounds. So these are our friends. People call them probiotics. We usually don't know how they work. But Lactobacillus bifidobacteria, these are commonly found in yogurt. Helicobacter pylori, I told you about earlier. But there are hundreds more that could be listed here. Many of them, we don't even know what they are or what they do. This, of course, is yogurt. Yogurt is good for you. But you have to keep taking it. Okay? I can take some lactobacillus, gets in my gut, but it doesn't survive very long. One thing we would like to do is to figure out how to introduce good bacteria that we know are killing some pathogenic organism and actually have it stably integrated into the gut bacteria. So it would be like having an antibiotic, except the antibiotic just kept producing. You took it once and it kept you safe. If you've got a bacterium that can protect you, then it would keep going. And I think this offers some wonderful opportunities for medicine. Okay, if ever we're going to make medicine cheaper than it is at the moment, if it was a medicine that involved a bacterium that could grow inside you, could produce compounds, or however it stops pathogens from growing, that would be a very good thing. I think this will come. We will figure out how these things work, and we will get there. Lactobacillus CUHK, this is one of the yogurt bacteria. I have to tell you an unfortunate story here. The company Nestlé's, who make excellent chocolate, they also are in the yogurt business. And they did some experiments looking at the kind of lactobacillus that you found in yogurt. And Lactobacillus CUHK was one of them. Another was called Lactobacillus johnsonii. They started playing around with one of these bacteria, one of the lactobacilli, and they found some that were much better at protecting the gut bacteria than CUHK or than the johnsonii. But it was a genetically modified bacterium that did this. And they had done it just at the time when Switzerland got all bent out of shape and the rest of Europe about GMOs. And so Nestlé's was so concerned that the activists would come and shut them down that they stopped working on this strain. Okay? And so we don't know what this strain might do, what it might be helpful for. I think this was one of the rather sad effects of the very early GMO movement. Here was something that looked good, but it got shut down. Now, I want to tell you a story about Clostridium difficile. If you go into a hospital and spend any significant amount of time there, there is a very good chance that you will be infected with Clostridium difficile. It spreads rapidly in hospitals, and many people come out of hospital with a C. difficile infection. And it's a nasty thing to get because most C. difficiles are resistant to all of the antibiotics that we typically use. Super resistant. So what can you do? Well, it turns out you can do something that sounds rather disgusting. You can take human feces from a healthy individual, that is shit from a healthy individual. You can mash it up, get rid of all of the sort of fibrous material, and make a little suspension of the bacteria. And if you drink it, it will get down through your colon, and it will cure C. difficile 95% of the time. There is something in healthy human feces that kills Clostridium difficile. And in fact, this is now used as a treatment in hospitals in the US. Sounds rather disgusting. Who wants to be drinking shit, as it were? Well, I certainly don't, but fortunately you don't have to do that. You can actually do it now by injections of one sort and another and by getting it other ways. And in fact, there's a lady at Harvard Medical School called, come on, okay, Betty Homan, and she came up with a way of taking these extracts from feces and actually making a concentrate of them and putting them into a tablet, a transparent tablet that you can take in much the way you take many drugs these days. It's just a little capsule and you put it in. And it turns out that when you do this, this works just as well as the old-fashioned method. People used to do this, just pour it down people's throats, which was disgusting. But this works much better. But she presented me with this slide. And of course, the possibility here is that because it's a transparent tablet, it doesn't look as appetizing as it might. This is a joke, though. This is not how they really do it. She also likes to point out that by taking a tablet or taking a capsule, you minimize the bad aftertaste that if you were pouring it down your throat, you would get. Now, it turns out that C. difficile is not the only pathogenic organism that can be cured this way. There are many other bacteria that live in normal human feces that can solve these sorts of problems. And the bottom line is, we do not know how any one of them works at the moment. This is totally unknown how they work, and yet here 95% of C. difficile cases are cured by this. And that's really important, because what used to happen was people would go home, because they wouldn't keep them in hospital forever, but C. difficile causes uncontrolled diarrhea. And so the people who had it never knew when they were going to have a sudden attack of diarrhea, and they didn't want to be on a bus or on a subway or in a car when it happened, and so they would just stay home. And I went down to visit Betty Homan, and she was showing me she's got a wall full of letters, cards, notes from people who've been treated this way. And there was one particularly touching one. It was from an 80-year-old woman who said for the last 8 years, she had been at home, couldn't travel anywhere. She just had to stay at home. And after this treatment, within a week, she was now free. She could go out. She'd actually started taking yoga again. She was so grateful. This letter was just fantastic. And you can't believe the kind of difference that some small treatment like this can make. And there are going to be hundreds of treatments like this once we understand how the bacteria that live with us keep us safe. Now, after all of this, one of the things that we don't know is how life got started on Earth. If we look back, we can go back so far. We can see that within a relatively short time after the Earth cooled down, life began to appear. One possibility is that actually life started elsewhere. Life might have started on Mars, because Mars is about the same age as Earth. There are constant meteor impacts, and we know that material from Mars makes its way to Earth. And what I'm hoping is that before I die, we will find out where life came from, and maybe we'll discover it did start on Mars, and we're really all Martians. Thank you. Happy to take any questions.
Let us again express our gratitude to Sir Richard for his wonderful lecture with a round of applause.
All right, now as usual, we can take several questions from the audience. But before you do so, please tell us your name, your department, and your year. Can I have
There's some microphones out there for people.
Yes, the microphone is just over there. Anyone want to? I think you all are beginning to love bacteria after the talk.
I guess we have no takers.
Question from the colleagues from the School of Life Sciences first, just to warm up. KB, I'll give you.
Rich, wonderful talk. I love bacteria myself.
Oh, good.
So do you have any, you mentioned about all those bacteria taking, shit to kill disease. Mm-hmm. Is there any, in your opinion, is it, okay, we understand we do not know at the moment. Any wild guess what could be, the cause?
Yeah, well, I'm sure, that what is happening is that every time food passes through your gut, the stuff you don't want makes it out into the feces, and of course it washes a lot of the bacteria that are normally living with you, it washes them through. And so feces turns out to be quite a nice zoo of bacteria, and a lot of people have been looking to see exactly what's there. And you can imagine many different ways in which they could attack C. difficile and stop it. It could be a direct interaction. C. difficile perhaps binds to one of these organisms, and that stops it growing. It may be that one of the organisms is producing some chemical that specifically targets C. difficile. And we already know that within the microbiome, that within your gut, there are organisms that are producing specific compounds that will attack individual other strains of bacteria. More interestingly, though, we know that the bacteria living in your gut put chemicals into your cells, and they talk to the brain. There are many chemicals, many indications that your gut, the bacteria, talk to your brain and affect brain function. So, for instance, there are hypotheses out there at the moment that things like Parkinson's disease are actually being caused by some problems within the gut bacteria. We also know that the brain makes more neuronal connections to the gut than any other organism in the body. And so the brain is being spoken to by these gut bacteria. I would love to know what they're saying. And I think here is a wonderful opportunity for research for anyone who's interested in this area. Much to be discovered here, and a lot of it, I think, is going to be very important for human health as time goes on, as we understand what is going on and how these bacteria do this.
Okay, student over there.
Okay, hi, sir. I'm a
Hi.
Year 2 medical student, and as I listened to your talk, I noticed that you have mentioned some current events like anti-vaccine movements and also like global warming deniers, and also I've noticed your stance on GMOs if I search your name on Google. Yeah, and I want to ask you, do you have any suggestions for countering scientific misinformations in society nowadays? Thank you.
Okay. Well, I mean, you raise a very good point, because one of the things that is certainly happening in society is that there are many people who are increasingly skeptical of science. If you'd asked 20, 30 years ago, I think most people would have held scientists with high esteem. They would have said they're good people, they listen to what you have to say. But what has happened, I think, is that the media has discovered there's mileage to be made by pretending there's a debate. Climate change is the classic example. Whenever they run a story on climate change, they try to find someone who denies it and then pretends it's a 50-50 argument. And this is just simply wrong. The same thing with vaccination. There was this story that vaccination caused autism. This was based on fabricated work that was done in London. It was soon discovered that it was fabricated. And yet, the anti-vaxxers, the people who are against vaccinations, continue to tell the story. They just never stop telling this story because it sounds like a good one. But in fact, they're doing tremendous damage. You know, in the US at the moment, there's great concern about measles. You know, measles is a disease that should not exist in the developed world. We have terrific vaccine against it. You know, it should have been eliminated. But what's happening is the anti-vaxxers have convinced small communities, oh, you mustn't vaccinate your kids. And it just takes one of them to get measles. Measles is endemic elsewhere in the world. You go to Africa, you go to many developing countries. Measles is a disease. And it spreads incredibly rapidly. The interesting point is that when a case of measles occurs in a community where they hadn't been vaccinating their kids, I will guarantee within a couple of days all of those kids go to get a vaccination. Okay? So the parents know that this is something that works. They go out and do it, and yet beforehand it takes a major outbreak before they suddenly decide they're going to start listening to science. But I think we also have some problems ourselves in that we do not communicate science terribly well to the general public. First of all, there are very few of us who actually make a point of going out and talking to the general public in language they can understand. You know, most scientists just resort to telling you about fancy experiments and using terms that the general public doesn't understand, very often that scientists in another field don't even understand. And I think we would do well to make sure that all of the scientists who we train in the universities and train earlier on in schools learn how to speak about their science to the general public. And I have a test that I give my students. I tell them, I call it the grandmother test. And the test is to be able to tell your grandmother what you do in the lab every day and tell her in words that she really understands. And to test whether or not she understands it, she has to go and tell all her friends how smart you are because you're doing this wonderful science in the lab. And I think this is a very good exercise, just to find someone who is not a scientist and explain either what you do or what somebody else does. And in this way, I think we can regain the trust of the public, but we've got to talk to them. We have to go out and talk to them. Go to local Rotary clubs. I go to Rotary clubs. I talk about GMOs at the moment, but I've talked about other things. I go to classrooms. I go to school. I talked to a class of 12-year-olds the other day. And afterwards, they all were told they had to write me a note thanking me for going and talking to them. One of them wrote back and said, well, her parents had been anti-GMO. They were terribly against GMOs, and I'd been spreading the pro-GMO message. And she said she talked to them about what she'd heard from me, and now they had become convinced that GMOs were safe. Well, I thought this was the greatest conversion I'd ever heard, and this just talking to a 12-year-old. So talk to young kids, because they're actually much more open to things than their parents. And they can sometimes convince their parents of what is right and what isn't. So I think here's some homework for all of you. Find someone who is not a scientist. Go talk to them about what you do. Okay?
Thank you very much for your response.
So the take-home message is to always talk to your grandma. It's easy for me because my grandma no longer lives.
Well, but, you know, you can go and still talk to her. It's just she's not listening anymore.
Talk to the photo. It's easy. Well, why are you so quiet? Any question? Good.
Thank you very much. So I want to add to the benefits of the bacteria because they are also symbiotic bacteria and also bacteria that can help the agriculture by interacting with the plant in the mycorrhiza. So could you elaborate a little bit so that the audience know better the benefits of the bacteria?
Sure. Well, you know, when we talk about the bacteria that live with us, we talk about them as the microbiome. And every organism that lives on the face of this earth has a microbiome associated with it. And in the case of plants, there are bacteria living in the soil that make nutrients, produce nutrients that help the plants to survive. Rhizobacteria, a good example. There are bacteria that live on the leaves of plants that can help the plants survive. They can produce compounds that will stop pests from eating the plants. This is a good thing. But in general, we have done so little work in this area of looking at the microbiomes of other organisms that we know very little about it. Our knowledge is minuscule by comparison with what's out there. One of the interesting things that I'm particularly interested in is soil microbiome and how you can persuade bacteria to regenerate soil. You know, if you look in many parts of China, there are major areas that are basically barren. Nothing will grow there anymore because of chemical pollution. It would be very nice to understand what sort of bacteria we could spread in these areas that would regenerate them, would remove the toxins, and would regenerate the soil. This is something that can be done. If you look at many areas of the world where the land is fairly barren, it's rocky, but, you know, rocks can be eaten by bacteria. Rocks can produce soil. I have a project that I'm trying to get started in East Timor. They have a major problem there. They don't have a lot of arable land, but they have lots of land that's rocky that I think could probably be rejuvenated and could be made into good soil fairly easily. And so the government in East Timor are quite interested in this. And of course, they don't have a lot of money, and so they will need some help to get it going. But I think there are wonderful opportunities here to deal with soil and to make soil much better. So, but every organism, all of your favorite organisms have a microbiome, and we can learn how to use it. I have one here. Maybe you want to keep this one up here and leave that down there.
Jolie, School of Life Sciences. I hope I'm not preempting your talk or your message on Saturday about GM food or GMO in general. What's your opinion concerning the future of GM human, for example? Where do we draw the line?
Well, so are you talking about plants specifically, or are you talking about human editing? Human, okay. So I'm sure you all know about GMs, and if you don't, you could come and listen to my talk tomorrow, or you can go to the website, supportprecisionagriculture.org, in which there are lectures that I've given on this subject before and a lot of interesting information. With plants, there are clearly many things that we can do to make them function much better than they do at the moment. So, for instance, with climate change, we're going to need drought-resistant plants. We're going to need flood-resistant plants. We're going to need plants that will be able to resist many of the pests that keep coming around. And, of course, whatever we can do to plants, we can do to other animals, too. So people are already working on farm animals and trying to make farm animals better than they are at the moment. But then, of course, you get to this controversial issue of human DNA editing. And it is quite clear that it will be possible, maybe not at the moment, but it will be possible to cure genetic defects by gene editing in the embryonic state. I think this is something that's going to happen. There are many diseases that one can think we can cure in this way, either by replacing a defective gene or just knocking out a defective gene. But we're not ready for it yet. And the problem was with Dr. He, who did these first experiments that caused all of the problems, he didn't get permission to do it. He didn't consult widely and let people know what he was doing. And what he was trying to do was stupid. You know, this was not the disease to go after. It's not even a proper disease. It was just plain silly. And I think it was because he misunderstood the biology. He was not basically a biologist. He didn't realize what was normal ethical conduct among biologists, and I think he thought that this was a way to get himself a Nobel Prize by being the first person to do it. And if that was his intention, then maybe he'll be the first scientist to go to jail for trying to edit humans, which would, I think, be a much better result of this whole thing. I mean, it was just plain silly. But we will learn how to do it. The reason it's silly is we don't know how to be precise with humans yet. With plants, you don't have to be precise, although most of the time we are. But with plants, if it doesn't go where you wanted it to go, then you can kill the plant, incinerate it, and find the plants where it has gone. You can't do that with humans. You can't take 100 embryos and then just select the one that happens to have picked up the mutation that you want, because that's just not practical. And so I think my guess is within 10 years, we will know how to do this precisely, and we will be able to solve some common diseases. But we need to do it transparently. We need to make sure there are rules in place and that the people who do it behave in an ethical fashion and make sure everybody knows about it. Then I think it will be okay.
Well, Professor He won't be the first scientist to go to jail. I know a chemist who made LSD in the lab. He's still in the jail.
Yes, there are plenty of scientists, unfortunately, who went to jail.
There's a student over there. Please start.
Good afternoon, Sir Roberts. My name is Vincent, and I'm currently year 4 undergraduate majoring in chemistry. From the biography, it was told that you got your PhD in organic chemistry, and for most of the organic chemists, they were focusing their career on new methodologies or novel reaction or finding new synthetic route of natural compound. Is there any particular reason that after your PhD you want to shift your career from the realm of small molecules to macrobiomolecules or even the realm of bacteria? Thank you.
Yeah, there's actually a very simple reason, but I'll put it in the context of a lesson for students. So when I was a kid, I read this book. It was entitled How I Became a Detective, a typical police detective. By the time I'd finished that book, I knew I wanted to be a detective. This has happened to me many times during my life. I've read a book, it sounded absolutely fascinating, and I thought, this is what I want to do with my life. When I was finishing my PhD, I actually had all the results I needed in the first year, but in England you had to spend 3 years. So my second year of the PhD, I was just doing experiments that were not particularly clever, and most of them didn't work. But I spent a lot of time over in the university library. And during this time, I discovered there was a subject called molecular biology. There was a journal of molecular biology. But there was a book written by John Kendrew called The Thread of Life, which described the early days of molecular biology at the MRC lab in Cambridge, at the Pasteur Institute in France. And by the time I'd finished that book, I knew I wanted to be a molecular biologist. And this one stuck. And so I went and did a postdoc in a lab where I could do some molecular biologist, and I still do. I also loved math and puzzles and games when I was a kid. And now what I mainly do is bioinformatics. And bioinformatics is being a detective, it's doing math, and it's doing molecular biology. And so I've managed to combine all of the things that I've been passionate about during my life in this one research endeavor, which I follow at the moment. And I would advise everybody, if you're doing something, don't feel you've got to necessarily only do that because your parents said, oh, this is the thing, you know, you should be a doctor, you've got to do this. If you read something or find out about something that is more interesting than what you're doing, do it. Go off and do. Don't be afraid to change. Don't be afraid to fail. You know, failure is the best thing that happens to students. You may or may not know this. Every time you do an experiment in the lab and it doesn't work, there's usually 2 reasons. One reason is that you screwed up. You know, you dropped the stuff on the floor or you weren't careful enough. But every so often, nature is trying to tell you something. Nature is trying to tell you that the reasons you did this experiment were wrong. I love it when students come to me and said, well, I did what you told me to do and it failed, you know, and I say, oh, go back, do it again. But if it keeps not working, there's probably a discovery to be made. This is the most exciting thing that can happen to a research scientist. So never be afraid of failure. Failure is how you learn. The people who don't fail learn almost nothing. So I have a hard time with teachers who are always telling their kids, no, you did bad, you only get a D because you failed at this. Failure is a good thing. You can learn so much from failure.
Thank you very much.
If my memory serves me correctly, I think the professor of organic chemistry of Sheffield was Professor W.D. Ollis.
Ollis, that's correct. That's who I did my PhD with.
Ah, yes. He was very inspired.
He was.
Inspired.
Yep.
Very good. There's another one. Another student, please.
My name is Christopher Wong. I'm a year 3 psychology student. My question is basically, I recently watched a video about industrial farming, and actually that has caused a very large-scale loss of the biodiversity of bacteria in the soil, and that is actually going to lead to a lot of large-scale food shortages over time, and that one way to recover is to perhaps help the soil regain its bacteria biodiversity. But with so much industrial farming we have on Earth nowadays, how could we recover so much soil in so little time before we all, starve to death?
Okay, so when you're talking about industrial farming, are you talking about growing crops, or are you thinking about animals?
Crops, because as I understand, the chemicals that we use is actually killing a lot of the bacteria in the soil, and over time we're losing more and more arable land.
Well, good agronomic practices take care of the problem. So typically the way farmers used to do things before they were doing things on the large scale at the moment is you would leave the land fallow for a year. You just let it sit there, don't try to put any crops on. It also turns out that if you alternate crops, you grow one crop and then the next year you grow a different one, this can help the land. And if you talk to the really good agronomists, they know how to keep the land in good shape. But as we learn more about the bacteria that are in the soil, I'm sure we can find better ways of doing it. It will just lead to yet better agronomic practices. And so while GMOs can help, they're not the whole story. They will give you better yields, but you need also to remember that it's important when you fertilize them, when you put water on them, the plants that you grow, and so on. And good agronomic practices can do wonders for any community. Okay?
Thank you.
I should say I'm not a plant biologist. I'm just reading and talking to people about all this stuff.
My colleague already showed me the sign that we have to stop.
Okay.
So now, after listening to Sir Richard's very inspirational talk, I think we should please all join me to thank Sir Richard once again.
Okay. Thank you. Thank you.